Hello, everyone. Appreciate you stopping by our mini presentation. My name is Vincent Trong. I'm going to share our work on comparative transcriptome profiling of human iPSC nociceptors to primary human DRG tissues. So the pain drug discovery field has had fairly limited access to relevant human cellular models. The nociceptor is the cell type important for pain transduction. You can get these from donors, isolate their primary DRG, purify them, but there are a number of drawbacks, which include donors are very hard to come by. There's heterogeneity between donor to donor and also the isolations. And you also have a very short assay window as there's a loss of function within the nociceptors in culture. With human iPSCs, you can also derive these from donors, perform directed differentiation, make these nociceptors, and this gives you the benefits of being fully scalable. You get consistency with large batches, and it works on multiple different donors, so you get a genetic background as well too. For this experiment, we performed 3 different differentiations from the same iPSC line. Within 24 hours, we go through a primal ectoderm population, and we can pattern that to a HOXB4 expressing neural crest. And within 7 days, get an immature sensory neuron that expresses islet1, BRIN3A, peripherin, beta-3 tubulin. For this experiment, we focused on the maturation throughout 4 weeks. We took weekly RNA samples and performed bulk RNA sequencing. And the big question we wanted to answer was how similar our nociceptors were to primary human DRG tissues and as a control, human iPSCs. So the first analysis we ran was a principal component analysis, looking at the largest sources of variation. You can see the primary DRG tissues right here. 3 of them are from patients with pain. 3 of them are from patients without pain. Here are the human iPSCs and the iPSC-derived nociceptors. And they are different. Primary DRG contain a lot of different cell types, Schwann cells, fibroblasts that make them different. But what's interesting is over time, you can see that the iPSC nociceptors trend towards the primary DRG. And if you look at the PC2 second source of variation, the iPSC nociceptors and the primary DRG are very similar in comparison to the iPSCs. So next, we wanted to look into DRG-specific factors. Ted Price out of UT Dallas has done an analysis on multiple different tissues and come up with DRG-enriched genes that are conserved between human and mouse. Of the 173 conserved factors spoken in the paper, 90% of the genes are expressed in our cells. The majority missing are related to myelination or immune cells, which makes sense because we make a pure population of neurons. You can also look at heat map gene expression of the iPSCs, the weekly time points, and the primary DRG by gene families. Ion channels are very important. There's piezo2, calcium channels, TRPV1, also neuropeptides, GPCRs, and cell adhesion molecules. Everyone has their own target of interest, which is why we created the real DRG and ID interface, which you can look through our RNA sequencing data via our website, type in a gene of interest that you have, and come up with a gene expression plot so that you can understand whether your gene is expressed and the optimal time to assay is. Common pain targets are sodium channels and the TRPV1. This is just an example of the real DRG and ID application. You can see after 1 week, NaV1.7 is strongly expressed. NaV1.8 is also expressed within a week. We use a RLAG value of 6, NaV1.9 is above that cutoff, and then TRPV1 is expressed as well too. We also wanted to look into what subtype we were making within our neuronal population. Ted Price also has another paper looking at the gene signatures of 10 human DRG neuronal subtypes. We took the gene signatures from that paper, analyzed via the RLAG value, and from this concluded that we have a mix of A fiber population and many C fiber subtypes. Lastly, we wanted to look at the time course maturation and perform a goal enrichment analysis. We wanted to look at several key pathways related to axonal, neuropeptide, synapse organization, and how they related to one another, and then also the genes behind them. With that, we have a scalable and consistent iPSC-derived DRG nociceptor population that is important for pain drug discovery. They express a lot of the same markers that real DRG neurons express. And with that, we want to thank you for visiting our poster. Big shout out to Drs. Ed Price and Pradipta Ray for providing the human primary DRG datasets. If you have any more questions, feel free to reach out via email, and have a great rest of your ISSCR.